says we're streaming.
All right.
Yes.
Welcome to the Sugar Science. We are streaming live with dkNET. Welcome, dkNET. We have with us today Maryann Martone, PhD. She's the principal investigator of dkNET. Jeffery Grethe, PhD. He's also a principal investigator of dkNET. And Ko-wei Lin, PhD. She's the project manager of dkNET. Welcome. Thank you so much for joining us.
Thank you so much for having us. We're really looking forward to this.
Yeah, it's gonna be great. We would like to just sort of walk through with some Q&A here. And I wondered, Maryann, if you could describe the history and role of dkNET.
Sure. dkNET was started in 2012. I think Ko-wei is going to share the slide and our web address with you. And it is a project of the NIDDK, the National Institutes on Diabetes, Digestive, and Kidney Diseases. It was established because NIDDK, like much of NIH, was investing a lot in research resources. By research resources, we mean the tools, reagents, organisms that people need to use in their research. And they established several different centers that were organized around different themes, different types of resources, but connecting them all together and making sure that people understood that these centers were available to them is a challenge. We know in the information age, it's not that we don't have enough information, we have plenty. It's just knowing the information that's out there that really is the challenge. And that's what dkNET was established to do.
Yeah.
And it's why it's called the NIDDK Information Network. So our little tagline is connecting researchers to resources. Now, most of our focus, of course, is biomedical resources that are relevant to decay research, as we call it. But of course, everyone knows that the body doesn't have simple boundaries like our NIH institutes. And so we really try to look across the whole biomedical enterprise and bring things that are likely to help people's research into an easy to search and centralized location.
Yeah, it's a fabulous idea to get a sort of the one-stop shopping for tools that type 1 diabetes researchers would need. I just wanted to ask, which brought each of you to dkNET? Maryann, did you want to go first?
Sure. My background is actually in neuroscience, so it's not that I set out desiring to create a dkNET, but Jeff and I started something similar in neuroscience back in 2008 called the Neuroscience Information Framework. And neuroscience, NIDDK serves the diabetes, digestive diseases community. Neuroscience has 16 institutes that fund neuroscience. And so they said, we have the same problem. We're investing in all of this tool development in online databases, and we don't know what's there, and we don't know how to find it. So we started cataloging essentially biomedical resources back then. And when the announcement came out for dkNET, we said, look, we already have an infrastructure for this. This is something we're very passionate about. We've done this for a while. Does it really make any sense for us not just to stay inside of neuroscience, especially considering how broad neuroscience is?
Yeah.
And could we not branch out? And so that's what we did.
Yeah, that's fantastic. Jeff, do you want to weigh in there?
Yeah, and my background is also on the neuroscience side, and mainly though on the informatics side. So I've had worked on a number of projects that have developed data repositories and have worked with data. And as Maryann said, we began working together on a number of projects, and dkNET really allowed us to take what we've developed in other domains and apply it in the DK domain, to provide these resources, to researchers.
Fantastic. And how about you, Ko-wei? How did you come to, participate with dkNET?
So my background is cell biology, and I also had some postdoc training in immunology, animal model, and biomedical informatics. So before I joined the team, I actually was working on a project to improve phenotype information retriever for an NIH database called dbGaP. And then during that time, I gained a lot of experience and communicate with a lot of bench scientists and computer scientists, and trying to learn what's their need and what's the gap between the biomedical science and the informatics. So that's how I gained an interest in bridging these two fields, and also about the data reuse and the data sharing. So since the mission of dkNET is to connect scientists with the existing resource and help them to find tools, reuse data, and also make their data fair, and then so their data can also be reused. So I found this project very meaningful and then can make biomedical research more efficient. So I joined the team and work with Maryann and Jeff since 2015.
Okay, great. Yeah, no, you have an excellent background that you're bringing to the role. It's almost like a perfect fit from what you've done historically. So fantastic. Jeff, can you talk a little bit about what scientists can gain by using the tools offered by dkNET? Maybe even like a little walk through the website, hit the high notes.
Yeah, so let me go real quick to sharing my screen here.
Yeah, there's 4 things I know that are on there that look, that we've heard from scientists that they use and that they are excited about is the resource reports, discovery portal, authentication report, and the hypothesis center. So yeah, let's.
So I hope everyone can see the screen. So yeah, so there are the 4 sections.
I can't see it, Jeff.
Oh, you can't?
See if it's working. There we go.
There we go. Perfect.
Sorry about that. So yeah, so the 4 main sections, and what I'll dive into are the resource reports and discovery portal. But we also provide, authentication reports and to enable researchers and provide them assistance in developing authentication plans, something that's being required by NIH. And then we also have the Hypothesis Center, which is an environment for people to explore omics data, without needing a lot of bioinformatics expertise. And this is something that Neil McKenna, I think, will touch upon in a future SugarScience event. But let me get into the resource reports first. And so the resource reports on the website provide information on specific research resources that scientists may want to use. So for example, if I go and click on the antibodies, and I've preloaded pages just because of internet, you get a listing, for example, of antibodies that may be of interest to you. If you actually click into an antibody, it will give you, a full report, the information, from, the vendors. We also have information on usage and citation information. So for example, where this has been used in the literature. And so this builds on our usage of research resource identifiers.
Yeah, that's great.
Basically uniquely identify the resources being used in publications, and then you can go into the report further. And we also provide, for antibodies, also validation and information, for example, if an antibody has been found to be problematic. For the discovery portal, this is an area where we bring in hundreds of different biomedical databases, information resources, and allow people to search across that. And so, for example, if one were to go in and do a search, you're then given a set of different categories for where we're pulling information from. We'll give you all the different databases that have that information. And then if you actually go into a specific database, we provide information about that, and then we also then link out to the database directly. So in a sense, it's we provide a lot of the metadata information through our portal, but we then direct you to the site that is providing that data.
Yeah, that's great.
So that's sort of my brief overview. And I will.
Can I just ask you regarding the Hypothesis Center? Can you just say a word on that?
Yeah, so the Hypothesis Center is really meant to be a space where a researcher can come, with a question, and then, that will actually be sort of directed to, a set of tools, where they can explore, for example, the connection between, genes and signaling pathways, explore, phenotypes that one might be interested in, how does one then from that get to, a set of genes, and then get to, information again on, for example, pathways of interest that one might want to explore. So really trying to put together, a set of guided tutorials, walkthroughs where we pull together different tools and sort of guide the investigator through those steps. And again, this is something that we're building out now in dkNET, and so sort of the first version of the new hypothesis center, will be up and running, in the next few months.
That's going to be fantastic. And I think an incredible resource, especially for early career scientists, when they're starting to develop their hypotheses and really do some deep dives into literature and other resources. So that's really fun. I wondered if, Maryann, could you do a case study, sort of like the use of dkNET led to blank? What have people been sort of, what are some of the things that people have been using dkNET to do?
Yeah, it's hard to sort of just do one because, as you can see, dkNET is very broad.
Yes.
I would say one of the biggest impacts dkNET has is in the area of these resource reports and these research resource identifiers. So these are the unique IDs that are supplied by authors to uniquely identify the tools and reagents that they use in their studies. And before dkNET fielded the infrastructure to do this, if you read your average paper, it was less than 50% of the reagents and tools that you could identify. Now in journals that support RIDs, it's up to 90 some odd percent. And the good news is that RIDs appear in over 1,000 journals, including all of the major ones, with Nature just coming on board, but Cell Press, PLOS, and others supporting the RID initiative. That's really what allows us to create this really unique resource, because we can use that information to pull information across it. So we see, like with a lot of resources like dkNET, we don't necessarily know how people are using it. As we often say, people don't cite PubMed in their papers when they use it.
Yeah.
We know that people are using it. We get thousands of visits per year. And we do occasionally see papers where someone said, I got my RIDs at dkNET. We're also happy, though, that we see evidence that people are using it to find resources. So they said, I found this tool using the resource catalog. I think the Hypothesis Center and the tools there are, of course, coming out with the more exciting things that we think of. It's really necessary to be able to find these tools, but of course, one always likes to know that you're impacting science. And so, as I think you may hear in a future webinar with Neil McKenna, or a presentation with Neil McKenna, even in the area of COVID and some of these, very rapidly emerging areas, he's shown how the Signaling Pathway Project can come in and help predict likely targets of COVID. And the interesting thing about his work with his O-minor tool and his transcriptomics tools is that he clearly saw, for example, that there was a sex difference, that there was an effect of progesterone on suppressing certain things in COVID. It predicted other types of targets like the ACE2. So I think that the platforms are very, very powerful, and being able to use those to promote science, I think, or actually do science, develop hypotheses, are starting to come. I actually see quite a bit of use of SPP in the literature. And I think Ko-wei is going to talk about our Summer of Data program soon, where we host students. I think at least 2 papers have come out where they've used the SPP or dkNET tools in their research. So one of them used training and fair data to create a new standard for representing glycosylation reactions. And then another used SPP again to predict a target. So we are seeing that dkNET is very directly supporting people's science.
Yeah, no, certainly. And it does seem that we're looking forward to hosting Neil in the future and having him sort of walk through his process, because in essence, it's kind of like a case study, right? He did a case study using COVID, using the tools to study COVID, and our audience might be very interested in how he did that, and maybe applying the same approaches to type 1 diabetes research. So that's going to be exciting. I wondered if Ko-wei, could you talk a little bit about the new Investigator pilot program in bioinformatics 2021 and the Summer of Data student internship program?
Let me share my slide first, so it will be more clear for people to get some link information. So we started dkNET new Investigator pilot program in bioinformatics last year, and this is our second call. The goal is to encourage early stage and a new investigator to apply computational or bioinformatics approach, such as mathematical statistical method to research.
Oops, we have a little reverberation.
Yeah, I think once you started to share your slides, Ko-wei, it kind of garbled, so maybe stop sharing and that might be better.
So should I stop sharing?
Yeah, I would do
Yeah,
that.
we can direct people to look.
Yeah.
You can just talk
You can
about
go
it.
to our website
Yeah.
or contact us. Yeah.
Okay, so
Oh, she's back. It's okay. Keep going.
so goal for this program is to encourage early stage or new investigator to apply their computational or bioinformatics approach, such as mathematics and statistical method, to pursue a research question measured in diabetes, endocrinology, and metabolic disease. So this award can provide funding for preliminary study that can serve as a foundation for their future grant application and development for their grant. So last year we have 3 awardees working on different projects. One is on inter-organ communication, and another one on analysis of the TEDdy study, which is a diabetes study. And the third project is the award is working on phenotype prediction of diabetes. So the duration of the funding is maximum 2 years, and then the award is 150,000 direct cost, and then the application due date for this year's program is February 26, 2021. So if you are interested or you know somebody who would be interested in, please check our website. There should be information on the homepage. And another program is the Summer of Data Student Internship Program. So it's an online 6 weeks program, and we provide $1,000 award to students who are or who will be working in a research lab in NIDDK relevant disease field during summertime. So it's a 6-week program, including 1 week boot camp and 5 weeks weekly hands-on and discussion session. We meet once a week, like Friday or in the middle of the day for 2 hours for each week. And then students will learn dkNET tools and some free resource to apply them to their research. And also they will learn the best practice of scientific rigor and reproducibility and other courses, including good data management course, and then also open science. They will also learn how to write good protocols for their research and also share their protocols. We also have course on fair data principles. So the program in previous years was very successful. From student survey, students said they have learned a lot useful resource, especially some free resource that they never known about and can apply to their research, and they were more comfortable in performing reproducible science after the program. So our previous students have been using, like Maryann mentioned before, they use a dkNET hypothesis center to analyze data and then to generate some hypotheses for their research. And I think 2 of them are preparing for the paper to publish. And then they also, some of them using resource report to find a cell line antibody that they plan to use, and also check the condition of those resources before they plan their experiment. And then I think one student, they use a fair data concept to develop a COVID-19 relevant database. So it's a very
it's great. There's tangible outcomes.
Mm-hmm.
Mm-hmm.
Yes.
Yes, application deadline for this year is on April 9th. So there is more information on our website. You can also check. Yeah.
Okay. We hope to see quite a few of you apply this year.
Yeah. And then I would just want to ask, sort of just to summarize, Maryann, what are the, can you just sort of highlight once more, what are the benefits of dkNET use for young researchers, EC career people?
So I think you can see that actually dkNET touches on the entire life cycle, including grant opportunities that are really available to get you started. We maintain a database that has also other pilot opportunities. It supports you developing hypotheses, looking for reagents, looking for other data sources. And I think that, a very interesting thing was said to me by an NIH program officer when he saw dkNET. He said, we have to break out of this mindset that we ask the person next to us what the best resource is to use. So you hear about this a lot where people say, well, I use that animal model because that's the animal model that was always used, but we know that that's actually led to some problems. For example, there's a famous set of neuroscience experiments that have all been done on an animal model that has in hearing, They used it as a model for hearing, only it is prone to congenital deafness, so it might not be the best actual model. So the idea that you can come to a location to actually choose deliberately and through thorough research of what tools do I need to use to perform my research, I think is a really important one. And I think training students early on how important it is to manage the research process right from the get-go, both in terms of the tools you're going to use, what are you going to do with all this data. New data sharing mandates come out, new rigor and reproducibility mandates come out, you're going to have to adhere to them. And dkNET really tries to help the new researcher navigate and use these things effectively.
Yeah, I mean, I would agree that it's a true template for the new way of doing research, and interfacing with bioinformatics is very important. But I mean, even just like going back to the slide that Jeff showed, where he showed, okay, this monoclonal antibody or this antibody was really a great one, and this was not as great. So then you have an informed choice.
Exactly.
It's not just you're calling somebody who used it in another paper and is and
that's
like,
right.
oh,
And just to very quickly, we know that with cell lines, they published a list in 2014 of cell lines that were contaminated.
Yeah.
Hundreds of papers were published with those same cell lines after that list was put out.
Yeah, so that's a fantastic stopgap.
Exactly. We did an interesting analysis where we looked at papers that use RRIDs where that information flashes in bright red compared to the general literature, and you saw a dramatic decrease. We don't know if that was the reason why, but it does tell you you need to be careful about what you use, and you need to do this due diligence, and we try to make that easy.
That's really great to share. I would also like to say that we've got, if I can share my screen for a second, I think I can. We have our resources available. I don't know if you can see it. We have our research portal on the SugarScience at www.sugarscience.org. And we've got second in line is the NIDDK Information Network. And we've actually seen quite a few people clicking here and going to use it. So it's very exciting. We also have other resources available, and those are increasing. So we encourage our listeners, our watchers to come for one-stop shopping to see all the tools available, including the most incredible NIDDK offering, dkNET. Is there anything else I have a couple of questions. Hold on one second.
We'd love
Question,
to take them.
how does dkNET work with NIDDK?
So dkNET is a cooperative agreement, a form of a grant where NIDDK actually has some oversight of the resource. So we meet with NIDDK monthly, and we have them on our advice. They serve as sort of a part of the steering committee of this project. And that's how we're able to very tightly couple ourselves to what's going on in NIDDK. So for example, that bioinformatics program was something that is obviously it's an NIDDK funding, but it's done through dkNET. So we work together very closely. We get recommendations, and they are very much involved with how dkNET is run.
Some of these you already answered, but what is the most used feature on dkNET?
Well, we always like to say we have a newsletter, and anytime it starts off with funding, that's always the most used one. So people are always interested. But overall, it looks like the resource reports and a discovery portal get the most usage on there. Jeff, did you have anything else to add on that?
No, I'd agree with that. And what we're really hoping is that, with the new rollout of the Hypothesis Center, over the next couple months, that will also be something that will drive traffic.
Right. And with the new NIH data sharing mandates, which go into effect in January 2023, we think that the FAIR data and data management section will start to get more use, but that's a little bit more modest. And it also, those two sections, the FAIR data and the Hypothesis Center, haven't been as well developed as the other two, but they're coming online fully this year.
Well, that's great. Here's the question. Does dkNET envision a partnership with T1D Exchange or Connect T1D in Canada in the future?
We are always open to these types of partnerships. Our goal is to, again, work with the full complement of resources that are available. And we have different levels at which people can get involved and get their resources integrated. But we would love everyone to become part of this information network. Both dkNET serves as one source, but we get our information from the rest of the community. So we would certainly welcome a partnership, and please feel free to contact us.
Great.
And we've also worked with various groups also in terms of, for example, with the research resource identifier, sort of helping them to onboard. So we did that, with HERN, with the biosamples, to sort of work through with them, how to get, some of those resources sort of integrated with our IDs. So
right.
Yeah, I think that's, it's fantastic that you've got this template and you can standardize, I guess, in a way,
Yes.
all these disparate organizations, which is fantastic.
And that's really been one of our goals, even for things like protocols. Everybody has their own way of putting them up there. But when you standardize them a bit, you can only standardize so far in biomedicine, and we're very practical about this. But when you standardize a bit, it really improves the flow of information across these resources. So we really do like to work with groups and new resources coming online and just say, look, here are some options for you that if you implement them, life will be a lot easier for everybody.
Yeah, no, it's fantastic.
Yeah.
And here's one more. What is the most powerful aspect of bioinformatics for scientists studying T1D currently, in your opinion?
Well, that's a big question.
Yeah.
So I have been very impressed. I mean, one of the things that has really motivated us, and we didn't have time to go into the open science aspect of it, but when we've seen the limitations inside of so many biomedical domains of the small study that tends to be underpowered and can only in an only very limited way sort of sample the full complexity of something like T1 diabetes or any biomedical disease. And I don't believe that without some sort of informatics, some sort of reasonable comprehensive data platform, we're ever going to quite be able to grapple with it. It's not that the simple questions have been answered, but as I like to say in neuroscience, our big triumph was Parkinson's disease. Well, they don't have dopamine, so let's give them dopamine, right? They don't have insulin, so let's give them insulin. But I think we've kind of run into the limits of that sort of thinking. These are complex interactions that go on. And with complex interactions, you almost have to have some computational approach. So I've been very impressed across domains of those that come together and recognize the minimal standards, minimal requirements that you share your data. You can take these small studies and you can concatenate them. Neil shows this very beautifully. The only reason that SPP works is he's got all these small studies that he can then look across and start to build consensomes and other things that look at all the data. And I really think for type 1 diabetes, type 2 diabetes, all of these, right? These are the types of approaches that are going to be required. The good news is they have a place for everybody as long as people recognize that their study is value. Your insight is important, but your data is really important. And if these data can get out there in a fair manner, I think it will open up an entirely new avenue for people studying these diseases, which have been so intractable in the past.
I agree with that completely. I mean, I think some people are, speaking of Parkinson's, you've got the Okamsi razor there, the Parkinson'some that
Right.
that they're building, where they're layering the different disciplines of data and looking at that from an informatics standpoint. And because type 1 diabetes is so multifactorial and you've got many researchers in many different disciplines, this could be an ideal place for it to come together. One more person said, do you interface with CPATH? That they're the consortia that's trying to make sure that the clinical trials are powered correctly so that they actually work.
Yes, no, exactly. We interacted with them a couple of years ago when they were doing One Mind for Research, but we haven't since. So one area that dkNET knows it needs to branch out into more is in the clinical space. We've really targeted the basic research, but I think all of these efforts need to be promoted, understood, and again, communicated, because we really are in a reorganization phase, I think, for biomedicine, where we're really now starting to pay attention to the fact that if we're going to do these trials, this is not just for the glory of the scientists, but because we actually want to learn something. And therefore we have to spend a lot more time thinking about statistical power and other things that traditionally, again, they get taught in one class early on, and then they tend to be forgotten. And that's really, as we're seeing, no longer acceptable. So that may be a good group to reach out to again.
Yeah, I'm sort of thinking just sort of on the
Yeah.
fly that it would be really fun to do an off the record, just a panel meeting with your team and maybe dkNET, you know, type 1 T1D exchange, T1D Connect out of Canada, and the CPATH, and just a little consortium, and you can exchange some ideas and brainstorm. So yeah, we'll reach out.
That would be a lot of fun.
Yeah. Thank you all so much. This was really informative, a very casual and fun way of getting to know what the offerings are at dkNET. And frankly, there's a lot there. And I would encourage our scientist audience to reach out and start taking part in it.
Yes, please do. There's all kinds of ways you can connect. And please let us know what you think. You know, the more feedback we get, the better we can make dkNET. So we always welcome suggestions and feedback, because we know there's plenty we could do to make it better.
Thank you, Maryann, Ko-wei, and Jeff, for talking with us today. And thank you for the great work you're doing.
Thank you for having us. We really enjoyed it.
Yeah.